Okay, so we left off on this slide starting our exploration of the cellular attributes of muscle and trying to figure out how muscle works. And we do have to be very much focused on the cell. So I say just here, just to let you know, they're about 100 microns in width and up to 30 centimeters long, especially in something like the sartorius, which is this great muscle you're going to see in your thigh. Okay, so very long cylindrical cells. So when we look in detail at one of these, right, myofibrils, right, take it out and zoom in on it, and we see, again, it's surrounded by the sarcoplasmic reticulum. We notice, again, this distinct pattern, and it's kind of hard at first because we just have a short piece of this. But this is what causes that really dark light striping that you see here, and if we go back to here, okay, that's due to this. So again, this is not as high a magnification clearly as this is. So within this myofibril, which is made up of myofilaments, mostly, right, myosin, which is thicker, denser than actin, all right, which is why they show it as a dark, thick purple, and then this, again, lighter, thinner line. So mostly actin and myosin with a variety of a few other supporting ones, and we'll talk about a few of those. And so we're going to focus in on this zone right here in one myofibril in a whole, right, of many in the cell itself, okay? And so all these myofibrils would have the same pattern, and they'd all be about in the same place, the actin-myosin organization, okay? So we're going to look in detail at this area right here, which defines the contraction and is called the contractile unit. So that's the sarcomere, because mere means measure, and again, sarco means flesh. So the sarcomere is the unit of contraction, all right? And it's very highly organized myofilaments that give this appearance to the sarcomere, okay? So the sarcomere zone or unit is really from one Z disc to one Z disc, and these letters are used. These are actually abbreviations for some German terms that were given when this was first understood and described through electron microscopy. So the Z discs do have this appearance, and so you kind of get this kind of zigzag, which might help you remember it, right? Z disc or Z line is fine. And so a sarcomere goes from one to the next, and the challenge will be defining this on your models in lab, all right? Now, within a sarcomere, there are a couple areas that we have to pay attention to because they're going to help us understand contraction. So we have an area in which the thick myosin and the thin, right, actin overlap. So we see this here from here to here, and that is called the A band. All right? An area that has only actin that you see here and is really from one sarcomere into the next, right, because these are, right, joined all the way down this really long cell, right, there again within a myofibril. Remember, there's many myofibrils in one cell. So this area which is only actin, which spans from one, right, sarcomere to the next, right, but is just really a fairly small area, is called the I band. All right. Now, the M lines and the H zone are within this A band. So an H zone, just to let you know, is really the space or area in which there is not an overlap. So this is, as you can see, myosin only. And then the M line, M does stand for middle, right? And this is created by a number of structural, again, proteins that help maintain this overall organization. And this organization is very important for a productive contraction. All right, if we take that sarcomere and just enlarge it here in a cartoon and then enlarge it again, this is what you get, right? So here they're just showing, instead of just a purple line, they're showing this as myosin actually is. It's kind of, it's kind of messy looking. It's got all these projecting, we just call these heads off the protein. So a very unique structure to this protein. All right, and then we zoom in on one specific head, and then so these are kind of, they're rod-like. I always think they look like golf clubs, honestly, but that's, it's all subjective, right? And if we look at this I band that is actin only, right? We zoom in on the actin myofilament. We see that actin is really, it's a dimer, so it's like two units, and it's two strands. Basically, people describe these as strands of pearls, and they do look like that globular protein strand linked together, and like you've intertwined two strands of pearls. But it is overall, it is a thinner place. Now, in this, if we zoom in, and we can see this on one of our models in lab, we see a couple other proteins we want to know, right? And then we zoom in a little more to really see those in detail. And that is troponin and tropomyosin. Now, tropomyosin is a rope-like one, so it spans the length here. Whereas troponin is actually 3 subunits, a smaller protein, that is sitting on top of this tropomyosin, right? Kind of at various intervals, you see that troponin then. Okay, so that's the overall structure, and it is a lot to know, a lot of new words, but please try, can approach it very straightforward, a little bit at a time, right? So elaborating here, I say that the myofibrils, which we're seeing one myofibril here, are about, a little over a micron in diameter, okay? There are thousands of sarcomeres in each one, and right, remember there are many myofibrils. If we said that a muscle cell has a diameter of 100 microns, you got a lot of myofibrils in there, right? Thousands of sarcomeres for each one. The sarcomere itself is about, 2 microns in length, right? So just giving you dimensions, you're not going to need to know that for the test. You will need to know the names of each of these myofilaments and some idea of what they do. All right, so to get a skeletal muscle contraction, now that we've seen all the players and the structure and organization, we want to figure out how this works. How do these muscles work together? How do they get excited? And how do they respond? And the response, again, is generally called a contraction, right? And do remember from our overall scheme of talking about, homeostasis and talking about the way we keep and maintain things in the body, we said your responders or effectors are muscles and glands. So here we're getting a look at a key responder, and its response, again, is contraction. It moves something, right? So it turns out, to get excited, we're going to look at electrical and chemical events. And this is really what determines any direction in the body. Direction, communication between cells, and direction of one cell of another cell is done through, again, electrical, which is going to be movements of charged particles, ions, right? Or and the cooperation of chemical, which could be a hormone, or it could be like a local neurohormone, neurotransmitter. So these are the directors, right? The getting excited, right? The response for contraction is that actin and myosin will bond, and they may, slide past each other. All right, so we call this, again, the sliding filament model. And it turns out when they do this, when they bond or form a cross bridge, and they begin to move, the myosin moves along the actin, that the entire sarcomere shortens. And we want to look at this together. So we said, there's thousands of these sarcomeres in, the myofibril, and, hundreds of myofibrils in a given cell, and, thousands upon thousands of cells in a muscle, right? So you pretty much see that once we begin this action of shortening on the very microscopic level, it'll result in shortening on the macroscopic, the big level, all right? So many of these things working together, we get a muscle contraction. And that would be an example. That's flexing your arm, abducting your arm, right? Extending your leg, stuff like that. All right? So we need to look closely at how we get that excitation going, right? Because this is key. You're not going to do anything. Muscle, again, isn't going to do anything until it is told to do something. So the direction comes from the nervous system, right? The nervous system is in control. So I say here again, neurons are the communicators, right? And here on one of your models, it nicely shows this end of a motor neuron coming down and making, getting very close, making contact with a muscle cell. So that's a really good model to, look and think about what we're talking about here. All right. So here's our end of our motor neuron coming down, and the end of this is an axon, and it's myelinated, and as it gets to the end, it branches, right? And we form these little kind of bulbs at the end of the axon. All right. So again, skeletal muscle is voluntary. So what controls it? Well, that means that your cerebral cortex, right? If you say something is voluntary, your brain, and specifically we're going to learn the frontal lobe, so which is part of the cerebral cortex, is going to be telling, and it's going to send that information as electrical stimuli, which will be converted to chemical and then converted to electrical again, okay, when we reach the muscle. So motor neurons, again, here's the end of this. We're going to look more at motor neurons in the next couple chapters. The neuromuscular junction, so joining between the nerve and the muscle, and here it is. We zoomed in and zoomed in again. All right, is also known as a synapse. So again, like we said, syn for the synarthrotic, we said that's a joining. So the synapse is a joining area. We will use language like presynaptic, so before the junction, and after. So presynaptic terminal is the end of the axon. The postsynaptic membrane is the sarcolemma, right? The synaptic cleft, again, is that physical space shown here. And then synaptic vesicles, well, these are what form in the axon and are released by exocytosis, into the synaptic cleft, and they diffuse from this area where they're high to this area where they're low, across that short distance, and they can then interact with a muscle. All right? So a motor unit, so we're defining a lot of terms here, so just try to stay with me, right? A motor unit is the single, right, motor neuron and all the muscle cells it makes contact with, right? So here again, this is making contact with a muscle cell, but this could branch and make more contact. So I say it's a single neuromuscular junction, but that means everything that the motor neuron makes contact with. And I say that varies from just 2, and of course you just see 1 here, but from 2 cells to thousands. And this has to do with our fine motor control and our gross motor control, if you're aware of what that is. All right, so this excitation contraction coupling, ooh, lots of words. Yeah, no pretty pictures on this, right? And as I said, there's lots of nice animations. Please look those up or use the links I've provided. Okay, so let's get a few rules here. All cells have membrane potentials. All right, so it turns out what that means is that there's a difference in the charge across the membrane. Okay, and I say it varies from a -60 to a -90 millivolts at rest. Okay, muscle and nerve cells can use this, right, potential to generate their own electrical signals that we call action potentials. And that's why we say they are excitable. And these action potentials travel in a wave-like pattern. All right? How do they do this? Well, it's through the controlling the movement. So we have to get back to the house, controlling the movement of ions across this membrane, right? Specifically, either the axonal membrane, as we'll look at more in the nerve chapters, but here the sarcolemma, which will create a current, right? So in order for contraction, which we defined as actin and myosin binding, and possibly the myosin moving the actin, right, or moving along the actin, I should say. So in order for contraction to occur, there must be excitation or stimulation. So we must do this. We must get ions to flow and generate an action potential. All right? In skeletal muscle, this always starts with the nervous system. So a skeletal muscle, in other words, does nothing without the nervous system telling it to. Right? So if that nerve is damaged, you will have paralysis. Right? If that neural network is damaged from the spinal cord out, you will have paralysis, right? Now, we're going to stop the video here because I'm almost out of time anyway, and because you really need to see this in small bites. And we'll pick this up on the next video.